Rob Fleischer, he's a senior scientist with the Conservation Biology Institute at the Smithsonian Institution, the National Zoo. He's actually here in Hawaii for a couple of weeks doing some field work for a current project. But the thinking was, Dr. Fleischer was a few years back was the research faculty at UH Hawaii Manoa, has been working in Hawaii for many years, and now works much more broadly on conservation biology issues and genetics, particularly with birds, but many other species as well around the world with the Smithsonian Institution. And he was here in Hawaii, and he was gracious enough to take us up on our request to offer.
Anyway, I've been studying Hawaiian birds and related topics and some Hawaiian plant work and a little bit of Hawaiian insect work now for over 30 years. And it all started back when I was at UH Manoa. But my interest in Hawaiian birds actually came before that. I was a boy bird watcher, and I was always going out and looking through field guides, and I bought the Peterson Western Field Guide. And in the back of that, there's several plates of Hawaiian birds, and I would look at those and I go, oh God, I got to get to Hawaii so I can see those. Well, when I finally did get to Hawaii, I found out that I couldn't see them except maybe in museum trays at the Bishop Museum because they were extinct, or they were so severely endangered you really had trouble finding them. And that's a sad story of Hawaii. We have beautiful, incredible birds, but so many of them are gone. And what I want to talk to you about today is how we can study those birds, some of them after they've been gone, and some while they're still here, and how we can try to halt that tide of extinction and actually do something to conserve Hawaii's birds. And I'm going to talk to you about it from the standpoint of introducing you to the topic, but then also talking about how we can use DNA to learn things about Hawaiian birds, even after they're extinct, and how we can use it also to try to develop ways to mitigate some of the things that are impacting Hawaiian birds even today. So, Birds of Paradise Lost, it's not going to be about birds of paradise, except that people view Hawaii as paradise, and these are birds. And so I'm going to start out with talking a little bit about the history of the birds, the history of the islands and the birds, and their extinction, and what caused that extinction. And then I'm going to talk about two ways that we can study the evolution of the birds using DNA methods, using molecular biology, particularly genomics these days, to look at the evolutionary relationships, or what we call phylogeny of the birds that are here, where they came from, what are their closest relatives on the mainland, how are they related to each other, how rapidly did they evolve in the islands here, and then talk about how you can use DNA to look at the history of populations over short and long term, using ancient DNA and also some other fancy statistical methods to try to tease out what's happened to the populations over time. And then I'll try to show some examples of how we can use these methods to help understand what's going on out in the field in terms of predation and competition that would impact the birds. And I'll finish off with with some of our work on one of the other big threats to the birds here, which is invasive malaria, an introduced malaria and an invasive mosquito that transmits it to the birds, which has had really terrible impacts on the birds. Okay, but to start with some background information, you all know that Hawaii is very isolated. In fact, it's the most isolated archipelago of any in the world. And because of that, it's been a tough place to colonize. And so a lot of things have actually not gotten out here. Most things haven't. And very few lineages actually do get out here of almost anything. There are no native land mammals and obviously no native reptiles other than sea turtles. But in terms of land reptiles, land mammals, it's just a couple of species of bats were the only mammals that made it here. And then in terms of birds, there's actually been a fairly low diversity of birds, but there's what's called low primary diversity. Not that much has gotten here, but there's secondary diversity that has evolved in place through the process of what we call radiation, which is really just a fancy way of saying speciation in sort of a flowering speciation. And I'll get back to talking about that in a moment, but I wanted to take a side step and just talk about the origin of the Hawaiian Islands and how that and the pattern of island formation can really help us in understanding evolution of the birds and other organisms that live in it. This may just be a reminder for many of you who live here and see it every day, but the Hawaiian Islands arise over a hot spot, which comes from the mantle, and it piles up on the bottom of the ocean, the lava, and this will build volcanoes or mountains from the floor of the ocean up to where they become subaerial, and meaning they're out of the water and now they're an island, and they continue to grow. And what's interesting about this process is that it doesn't just happen in one place and just stay there. It's what we call a hotspot archipelago on a conveyor belt. So this conveyor belt process is basically the Pacific plate has been moving in a northwest direction for at least the last 50, 60 million years. And it's moved over the hotspot. And as it moves over the hotspot, you get these volcanoes that come up and they become subaerial. You get islands forming, these huge shield volcanoes, and they build up to as much as 15,000 feet. Then they become vegetated and soils develop. And then, as they get off the hotspot, when they come off of it, when the plate moves them past it, they kind of break with the lava, the spike is no longer under them, and you start to get what's known as subsidence. The islands start to go down. That's because you built up all this weight on the crust of the earth, and the weight weighs the islands down, but also the crust is cooling when you get away from the hot spot, and that also causes less swelling of the crust. And so the islands actually subside very rapidly. Maunakea at 14,000 feet approximately, and then you go to Haleakala, and that's about 3,000 or 4,000 feet shorter. And as you go to the next island, but it's not just the subsidence; it's also erosion, and this causes the islands to diminish fairly rapidly in size, to where when you get to Kauai, which is estimated a little over 5 million years, you end up getting an island that's really only about 5,000 feet in elevation. And then after that point, there's a bit of a gap, and you come to the next islands that are mainly rocky islets, we call them. They're like Nahoa, just rock croppings that stick out of the water, and then ultimately they go under the water, but atolls develop around them. And then ultimately, when you get even further up, these things go underwater completely, and you get undersea mountains. So on this map here, you can see there we go. You can see this linear progression of islands, and the oldest islands are up here in the northwest, Kure and Midway. Beyond that, there's just undersea mounts, and these are all atolls here, and you get a few rocky islets down here in Nahoa and Necker, and then you get the main Hawaiian islands here of Kauai at 5 million, and Niihau at 5 million years, and then Oahu 3, and then the Maui Nui complex at about 2 to 1 million years. And then Hawaii here, it says 1/2 to 0. Those numbers keep getting revised as we get better estimates, but the Big Island is probably, depending on where you draw the line with Mahukona, which is this thing right here, and the older Kohala Mountains, it's probably no older than about 600,000 or 700,000 years that it's been subaerial. So it's a fairly young island, and we're on the young side of a young island here. So Hawaii is really the youngest island. We sort of give it a standard error of 500,000 years plus or minus a couple 100,000 years for that, since there is some uncertainty. But what this does is it really gives us a great system for estimating evolutionary rates and the rates of species formation, the rates of molecular evolution, rates of morphological change. because you can use these islands as benchmarks of time, and then you can estimate the rates of of evolution of the organisms that live on them. And I'll show, I'll talk about an example of that later. So very few lineages have gotten to Hawaii, as I mentioned. One of the premier ones in birds, I'll talk a lot about today, are the Hawaiian honeycreepers. Based on the fossil record, the historical record, and different taxonomists' viewpoints of what constitutes a species, we estimate there was about 54 species, of which there's about 18 left now. So they've really gone downhill in terms of extinction in the last few hundred years. In plants, you have, of course, the silver swords, which is one of the other species that's colonized and has radiated and diverged from its mainland relatives. The closest mainland relatives, they don't look like little shrubs. They don't look like these beautiful silver swords. And then we have things like the Drosophila, which is probably the king of radiation, with over 900 species, although there may be two different ancestors that colonized at that point. So what I was saying before, you get a species that colonizes, and you get very few of those, but because there's so many open niches, there's so much opportunity with all these new habitats forming, especially with new islands form, you end up getting a lot of speciation and a lot of differentiation as species come in, evolve new species, and these new species adapt to the new niches that are present. And that's where you can generate this secondary diversity in Hawaii so easily. And I'll talk a little bit later about some of the other radiations of birds in the islands too, some of which are now all extinct, but they're also very interesting. I want to just give an overview of the birds of Hawaii. We sort of put these into two big groups. Native land, I'm going to talk about land birds mainly, but I'll mention seabirds a little bit here because there are some endemic seabirds. But native birds and then introduced birds, which I'll just touch on briefly later. But in terms of the native bird diversity, I said it was pretty low primary diversity. The seabirds are a little bit different. They move around the oceans. They live on other islands, not too near to Hawaii, but closer to the mainlands. And they have very vast abilities to get around the ocean. So things like albatrosses and frigate birds will be pretty much all throughout the Pacific, and they'll be also on the Hawaiian Islands, mainly on the Leeward Islands above Kauai. And then we have some raptors. We still have the Hawaiian hawk here. There were some other hawks here as well that are extinct, that were probably related to the harriers. And there was also an eagle that was related to the white-tailed eagle of Eurasia that went extinct probably at least 500 years ago. And there were also a group of owls that were here that weren't the pueo and weren't barn owl, but they were long-legged owls that occurred in the island, probably took a lot of birds, as did the hawks. There were also these waterfowl, not just the nene and the koloa, the native duck, but also these things that were large and flightless. We call them Moanalos, and there's an example of a turtle-jawed Moanalo right there that lived on Kauai. These are all extinct, and they were flightless. And there was also a lot of rails in the islands. We're down to the moorhen and the coot, but there were other rails here, and many of those were also flightless. And there was at least two species of flightless ibises in the islands, so which is very interesting. We're related to the white ibis of North America, based on our DNA work. So those are the native birds that are not songbirds. Songbird diversity is extremely low in terms of primary diversity. There's only 6 lineages or 6 kinds of songbirds here. The honey eaters, which are the o'os and kiwea, crows, which include the alala, but there actually were several other, maybe as many as 4 other extinct species of crows in the islands and on other islands besides the Big Island. Flycatcher the alapaio, which probably is made up of 3 species, and for some reason doesn't seem to ever have made it to Maui. And then radiation of thrushes, including the omao, which is here on this island, and then there's one other extant species left on Kauai that's critically endangered, and the rest of the species that occurred are extinct. Way up on the leewards in the island of Nahoa, and also the island, formerly on the island of Laysan, is an old world warbler called millerbird, and these are related to reed warblers on the mainland, and but they're not very closely related to any of them, and they seem to be their own distinct kind of reed warbler. But they only seem to have lived on Laysan and Nahoa. There doesn't seem to be any record of them on islands down here. They may have been on other islands in that area as well, though. And then the king of radiations, as far as I'm concerned in the bird world, beats the Darwin's finches handily, are the honey creepers. And I'll talk more about those. Probably at least 55 species. Here's a sampling, thanks to Jack Jeffrey, of some of the Hawaiian honey creepers that still exist, including the Laysan finch up on Laysan Island, and also introduced to Pearl and Hermes, and somebody in the audience here studied those in detail for doctoral dissertation many years ago. They're only up on those islands, also very endangered. Here on the Big Island, the Akiapola'au, which is an interesting bird in that it has a dual-purpose beak with an upper mandible that's long and skinny that it can use to pry things out, and a lower mandible that's stout and sharp, and it can use it like a woodpecker to hammer and make holes in the bark. And so it does this sort of hammering, and then it'll reach in with the upper mandible like it's a tongue of a woodpecker to pull the grub out. It doesn't seem very all that easy to use, but they make a living at it. We have finch-billed forms, and one of the things I should mention is by irradiation and differentiation, we mean that all these species came from a single ancestor and evolved. And they evolved all this diversity in form while in the islands. They speciated, they evolved the differences in their beak shapes, in their body size and shape, how long their wings are relative to their tails, and so on. All these things are considered morphology. And there's also a lot of variation in color, as you can see, and in sexual dimorphism, how different the sexes are. And also in their ecology, what they eat, where they live, and also their behavior, and including their song. The song variation is as much as diverse within the honey creepers, which is one radiation, one group, as it is in all songbirds. This is an incredible radiation that occurred here. Look at all this diversity. The finch-billed form, Makihi, and the Eevee with its long decurved bill that it uses to probe flowers for nectar. The Hawaii kepa, which looks like a little canary almost. Maui alahio and the Hawaii creeper, which are both creeper birds. This looks almost like a warbler you would see in eastern deciduous woodland or over in Europe, but it's and when people first came here and saw all this diversity, and this is just a subsample here, I'll show you more in a moment, they thought that these belonged to many different kinds of bird groups. They thought that this was indeed a warbler, this was a finch, and some of these were in other groups. And so it took some good taxonomy work to figure out that these were all actually related and all part of the same group. And really not until the molecular era did we finally become convinced that these were all part of one radiation. The DNA really drove it home. Here are some of the specimens from the Bishop Museum. I think this is the Bishop one, yeah. Showing some of the extinct ones. And again, the diversity in bill shape, like the mamo here, which is another species that takes nectar, like the Eevee, and with a really, really decurved beak. They're extinct. Akialoa has a really long decurved bill also, but it uses it more like a wood hewer to probe into bark and get grubs and things. There's some really big finch bills, like the greater koa finch here, And then we have some things like the Maui parrotbill, which really has a bill that's very much like a parrot. And all of these things show tremendous diversity, as you can see. And if I was able to show you some of the extinct ones that we don't have museum specimens for, only subfossil bones, the diversity of bills gets even broader. There's things that my collaborator, colleagues Helen James and Storrs Olson have found that they're not quite sure what the bills could have been used for. They might have been used for fly catching, they might have been used for scooping things out of the leaf litter. And just the honey creepers have a tremendously diverse array of bill types. Okay, just going to mention introduced birds. Most of the birds you'll see around here are going to be introduced birds, in this area around Hilo and anywhere in the lowlands. And they're going to be things, as you know, like common minnows. What's amazing to me is the diversity of birds and where they came from. They're from all over the place. We even have birds from South America, North America, the cardinals, Japanese white-eye, and then things from North America as well here, like the California quail. A lot came from Asia. And if you count everything up that we think has been introduced, it's about over 150 species. And not all of them made it. I think right now the estimate is about 50 species are surviving in the islands. And some of them are flourishing. I think the most common bird is the Japanese white-eye in the islands, but house finches and cardinals and nutmeg and minnows are obviously doing well, as are zebra doves. So very successful with introduced birds here. Now, going back to the radiations, I said 6 lineages of songbirds and 16 of non-songbirds, based on our best guess right now from the DNA. Although there's some things that we don't have quite done yet with some of the fossil forms. Things that radiated well include the Hawaiian honey eaters, which is these two birds here, the Kioea, and this is one of the O'os from the Big Island, Moanalos. These are actually honey eaters from Australia and New Zealand, just put for comparison. And then I mentioned the honey creepers, and another one that I'm going to talk about is the Moanalos and the other flightless waterfowl. Okay, what happened to the birds? This is a nice graph that shows you, this is what our best estimate is right now, based on the fossil work that Storrs Olson and Helen James have done, and the taxonomy and the historical record from museum specimens of how many birds there were in the islands about the time before the Polynesians arrived. We estimate at least 107 species. When Cook arrived, we think that number was down to about 55, cut in half. And then a few years ago, it was at 31. We've lost a species at least since then. And of those, 11, actually I don't think it's 11 anymore, I think it's 9 now are listed as endangered under the Endangered Species Act. So Hawaii has really suffered a loss of numbers over a long period of time, and is still the capital of endangerment for birds and for a lot of other species. And what caused this? Well, people came and they depredated the birds themselves to some extent, and they probably caused problems, but they also brought things with them. Introduced predators like dogs or rats, and competitors, and things that could have been carrying disease like chickens. They also brought things that degraded the habitat like pigs, and later on like mouflon and cattle and so on. And they also brought, at least after Western contacts, we're not so sure about before that, disease. And that includes 2 diseases that have been particularly bad news for the Hawaiian birds, especially the Hawaiian honey creepers, and that's avian malaria and avian pox virus. And so these are the main factors that have led to these declines. And one of the things I mentioned before is with all the extinction and endangerment, we don't have a lot of birds we can go out and catch in the wild. And if we want to study their genetics or their behavior, ecology, you can't do that with the ones that went extinct, and it's very difficult logistically and for a number of other reasons to do it with the endangered ones. So for the DNA work that we did and started doing 20, 30 years ago, we had to develop special methods, and at the time they were just starting to develop these ancient DNA methods. And we got in on that early because that was the only way to figure out some of these things with these bones or with museum specimens. And that's how we've done a lot of our work. And it's not as easy as if you can get a blood sample from a bird or to do DNA. It's much more difficult because the DNA is very degraded and often contaminated with bacteria and fungus and so on. So it makes it much more difficult to work with and a lot less. This is just some examples of some of the things that Storrs and Helen have reconstructed. They reconstructed a whole view of Maui. Julian Hume, an artist from England, painted a picture of Maui that has all of the, I think it's 27 extinct species on it. So like a Moanalo here. You can even see an eagle over here, way up in the distance. This is one of the extinct owls that was feeding on some palila, and the nene, and then this other species that was very closely related to the nene, that was partly flightless, and then the giant goose, which was flightless. And then this is just an odd honey creeper that they're guessing was probably a flycatcher of some sort. The other thing I wanted to mention is, besides the work of Storrs and Helen and some of their colleagues in reconstructing the prehistoric avifauna, the historical avifauna was not really widely studied just after Cook's arrival. Cook brought some specimens back. There were a few other travelers that came and collected things like Townsend and DePay. But it wasn't until the late 1880s that a real boom in collecting in the islands happened. And this is because mainly of two people, Alfred Newton, who was a professor at Cambridge, an ornithologist, and actually his former student, Walter Rothschild, who was a banker and and a very wealthy man, but he just loved birds and natural history and had a huge collection. And both of them sent collectors to the islands. Alfred Newton sent Scott Wilson initially, and then later sent R.C.L. Perkins. And Walter Rothschild sent Henry Palmer, who came here and collected a lot of birds. And they were, what was interesting about that is that a lot of the species that we know that are in museum drawers now, we had no specimens of them up until that time. And they had found out, Newton had found out, and Rothschild, that the birds in Hawaii were starting to disappear for some reason. So they felt that they had to document what was here, and so they immediately sent collectors. In fact, Palmer, I think, was in New Guinea, and got letters saying, get on a boat and go to Hawaii immediately, and so he did that. They collected a couple thousand birds, and Henshaw also was part of that, and Monroe. Those birds are our only record, really, of I think at least 11 or 12 species that we wouldn't have any information on, honey creepers and of other species of birds without this action. And it seems terrible they collected these birds, but they were on their way out anyway, and at least we have a record of what was here. And what's really nice is Newton sent Perkins, who was not only a good collector, but he was also an excellent naturalist and observer. And some of our only information about what these birds were doing in the field come from his observations and the notes that he took. He was really he was a scientist way ahead of his time, really almost a Darwin in terms of his abilities. Okay, now I'd like to talk about some of the evolutionary relationships of a few of the groups here. I'm just going to talk about three in particular, although we've worked on most of the species in the islands that occur here, including extinct ones and extant. We'll talk about an interesting case of convergent evolution in these flightless waterfowl. If you go to Africa or Australia, there are these ratites. The ostrich in Africa, emus in Australia, you go to South America, there's rheas. You go to New Zealand, there's kiwis, but there also used to be moas. Moas are what the Maori called them. It means fowl. We call moas, we call chickens moas here. But moa meant, it was basically a big fowl. And and they were all related. They were all ratites. They were all related. When apparently Hawaii had its own version, but they weren't ratites because ratites got to where they got by floating with either continents or pieces of continents that moved on the tectonic plates after about 60 million years ago. But remember, Hawaii, no plates, islands coming up, nothing could have moved here that way. You had to colonize here. So what filled the niche of a ratite, and also to some extent of a grazing mammal like a deer, were large waterfowl. And these were known from fossils that were found on Kauai to Maui that were large flightless waterfowl that had very big bodies, large legs, and extremely small wing elements that were left, and almost no keel on the sternum. So that keel, like a keel in a boat, that's on your turkey or chicken, they had a very reduced one, almost flat, like ratites. So they really converged to that. And those were only on these older islands. And on the Big Island, there wasn't one. They've never found one stored in home. But they did find this large giant goose that they didn't know what it was. And it also had a very diminished keel and was flightless. It was very large. In fact, it was even a little bigger, I think, than the Moanalos up here. So we did some DNA work on these to find out what these really were and how they were related. And we found out that the Moanalos, the turtle-jawed one on Kauai, these ones here that are on Oahu and Maui Nui, were all related to dabbling ducks, like mallards. They fall out right in that group when you do a phylogenetic tree. And so they're all related. And when we did a dating, tried to date using the molecules, how far back it was that they separated from each other, they been in the islands probably for about 2 or 3 million years, and they became flightless on each of these islands. So they probably was a duck that came here, and it was able to move around for some time and colonize islands. And on each island, Kauai, Oahu, and Maui Nui, they evolved into these flightless forms. But Hawaii, remember, wasn't up until maybe 600,000 or 700,000 years ago, so they never made it there, because if they couldn't fly after that time, because they were already flightless here, and they couldn't really swim very well, because they'll sink. And so there was no Moanalo on the Big Island. But later, about maybe 1 million years ago, the ancestor of the Nene, which is a Canada goose, colonized the islands. And the Nene was able to get to all the islands. And on this island where there was no Moanalo, instead of a duck turning into a Moanalo, basically a goose did. And that's how you got the giant goose. So it's a really interesting case of what we call convergent evolution, where two different lineages with different ancestries, because of similar ecological pressures, evolved into filling the same niche. And the giant goose wasn't quite as advanced in that as the Moanalos were, but it was moving in that direction. Another case of convergent evolution is one involving some extinct songbirds. You've all heard of the o'os that were here in the islands. They went extinct in the 70s. The last one seen on Kauai. There was o'os on Maui Nui, Hawaii, Oahu, and Kauai, and there was the Kioer here, which was on the Big Island here. And they've always been assumed to be honey eaters because they look like honey eaters. They act like them. They take nectar from flowers, or they did. And if you look at their tongues, here's the tongue of the Hawaiian honey eaters, and these are the tongues of honey eaters that live in Australia and New Zealand and the Pacific. And so there was never any doubt in anybody's mind that these guys were honey eaters and related to these. When I started doing some DNA work from museum specimens on these several years ago, I couldn't figure out why they weren't falling out where they should, and I ended up deciding to do a gene called RAG1 here. And when I did that, I put it into a tree of RAG1 sequences that included all of the perching birds, all the songbirds and their relatives from the world. It didn't include all of them, but it included a sampling of, it covered the whole variety of them. And I expected the honey eaters would come out, the Hawaiian honey eaters would come out down here with their relatives from the South Pacific and Australia, and I couldn't get that to work. Instead, they came out up here with things like chickadees and swallows and nuthatches. And so this was really an unexpected finding. The honey eaters that looked and acted like honey eaters, had tongues like them, were actually related to cedar waxwings much more closely. So what probably had happened is some relative of waxwings, probably 12, 15 million years ago, had come to the islands, and in this place where there were now flowers that needed pollinating by birds, they evolved into that niche. Maybe they did it occasionally, but as they became more and more adapted to doing it, they developed these same traits that you find in the honey eaters elsewhere. So a really pretty astounding example of convergent evolution, and of how you can use DNA to uncover these kinds of things. So the Hawaiian honey creepers I mentioned are the king of radiations. You have all this diversity and form, these seed eaters and bark pickers and so on, and this wonderful image that Doug Pratt did. And we've been trying to work out the phylogeny of these for years. I did some of it myself years ago using small bits of DNA sequence, but it took getting a really good postdoc, Heather Lerner, to work on it about maybe 6 or 7 years ago to actually get somewhere with it. Because the radiation occurred very rapidly, and it was very difficult to get the molecules to sort it out, to sort out the branches. So I'm going to show you a fancy tree here. Actually, it's 2 trees, a cloudogram we call it. There's one tree here, and here's another one. And what we did was we sequenced the entire mitochondrial genome, which sounds impressive, but it's only about 17,000 base pairs. And we also sequenced about, I think, 8,000 or 9,000 base pairs of nuclear genes. And we did these analyses to build trees. And we did this from the honey creepers here that are extant, and also from these Cargeline finch, we thought were the relatives, and then some other songbirds. And when we put this all into a program that builds a tree based on the DNA sequence, we found out that, like we'd gotten before and other people had surmised, the honey creepers are actually related to finches. They're part of a finch, of the Cargeline finches, which include things like canaries and house finches and goldfinches and so on. But the closest relative to the honey creepers was actually rose finches from Eurasia. So the honey creepers, which a lot of people thought came from North America, didn't, based on what our findings, and I think these are pretty solid now. The honey creepers are actually probably colonized from Asia. And they were from a finch, a true finchville. So you think that the finches would be the ancestral state. But when they got to the islands, they stopped being just finches and they became warblers and parrots and all kinds of other things. And this is the same mitochondrial data set I showed before in the cloudogram, but this is just a tree, and it shows you in the tree the time frame, we think, based on using calibration points. So, as I mentioned, this is a rose finch outgroup here, and what we did was we took the ages of the islands and we found certain points like when Maui and Hawaii would have diverged, and we estimated that, we used that for that, an estimate of a 500,000 years from the age of the island, and we had another one over here between Kauai and Oahu. And when you put, and another one further up in the tree, and when you put those into this program that we use, it's actually called BEAST, it will allow you then to calculate a rate, and the rates were very constant, fairly constant, between the different calibration points, which gave us some confidence that they were real. Remember, if a bird can't colonize an island before the island is above the water. So we use that as sort of a maximum age of a split between 2 species. So if there's a species on island A and island B, if island B didn't form until 1 million years ago, that would define the upper limit to that time frame. They could have colonized after that, at any point, but we were using that point, making that assumption. And when we put those into the program, it gives us an estimated time frame for all the other nodes in the tree, all these other points where the branches split. And we found that the split at the base of the radiation was about 5.5 million years. So this whole radiation that occurred in the islands probably started when the time Kauai was coming up and was a big island, and there were still some other islands before that that were big. And then as the other islands formed, after they formed, the birds were able to colonize those islands. They were probably colonizing back islands. And every time they colonize, you get the opportunity, especially if it's a new island where there wouldn't be other species on there yet. If you colonize from Kauai to Oahu, when Oahu forms, you have an open space there you can colonize, and there's open niches. And so what probably happened is you're now isolated from your ancestors on Kauai, you're now on Oahu. You will then diverge, and you'll end up also maybe finding a different niche you want to move, I'm saying you want to move into, but something that you can start to adapt to move into. So instead of being a finch bill form, you start eating more insects, and your bills, over time, there's selection for your bills to get smaller and narrower, so it's easier for you to eat insects. And this is the process of radiation, of adaptive radiation. When you speciate, you're no longer able to breed or hybridize with your ancestor species back on Kauai, and you've now evolved into a new form, a new morphology, and a new behavior. And this happening over and over again in this tree is what leads to this radiation and to this tremendous diversity that came about in the Hawaiian hemisphere. Okay, I wanted to just say about how we can use DNA to look at population history and bottlenecks. Population history going over the long term, over thousands to millions of years, and also more recent time frames. And there's really sort of two ways, general ways. One is to use ancient DNA, so you can go back and look at bones that are 500 or 1,000 or 2,000 years old, and look at the genetic variability that occurred at that time point, and then you can compare it to another time point with other old specimens or to now. And so we've done that with a number of species in the islands, including Laysan finches, and also with Hawaiian petrels. Two of my students working on that, Taylor Calicrate and Andriana Welch, and then also on the nene. And I'll show you the example from the nene in a minute. The other way to do that is you can actually, with very sophisticated models that I don't understand completely, called coalescent models, you can actually take genetic variability from a sample of birds or from a single genome and use that. You look at the pattern of variability in that, and you can use that to infer what the history of the population was, because the population gets small, variability decreases, selection becomes less important, and what we call drift or random processes become more important. And these leave sort of benchmarks in the pattern of variation in the DNA sequences. And so I'll show you one example from that as well. The nene one is an older one, but it's really cool because we had found out that nene had very low variability, genetic variability, when we were studying them 20 some odd years ago. And we wondered if maybe that was because of the decline that they went through after Westerners arrived, after Cook arrived. So we looked at genetic variation in the mitochondrial DNA in nene, and it turned out modern nene had no variation that we could detect in the genes that we looked at in the mitochondrial DNA. They were all what we call one haplotype. We also looked at old museum specimens dating back 150 years or more, and we found didn't find any more variation, really. We found everybody there, 13 of them that we looked at, was the same as that one, but one museum specimen from Vienna was one base different in the sequence. So still, really, not much variability. So Helen James and Storrs Olson had collected a lot of nene bones, some from archaeological sites, middens, that date back about 500 years, and some from what they were calling paleontological sites, that were mainly lava tubes, that dated back, 3,000, 4,000 years. And so we were able to get DNA out of these bones. And we found that in the archaeological material, we didn't find any variation either. We found the same genotype. So going back at least 500 years, there seemed to be really low variability. When we went back further, and we had a lot of controls and to make sure that we were getting the right picture and this wasn't artifactual, we suddenly started finding a lot of variability. You can see all these branches here that come out to these other haplotypes indicate that there was a lot more variation 600, 800, 1,000 years ago than there was after 500 years ago. So the bottleneck we expected was in the 1900s. Instead, the bottleneck we found was back over 500 years ago. Something, probably the same thing that knocked out the Moanalos and the ibis and the rails, all at that same time frame, and the eagle. It happened in maybe from 600 to 400 years. A lot of species drop out about that point, and there was a peak in population size in the islands of Hawaiians at that point. Something caused a die-off at that point. The Nene probably went through the same thing, but somehow they didn't go extinct. They recovered and they came back because they were fairly numerous in the 1800s in Hawaii. But they also used to be on the other islands, and they didn't occur there historically, but they were based on fossils. So, so this shows you the power of the DNA for really giving you a more accurate history of what happened. I like to think, though, that maybe the reason why the Nene bounced back is because some chief on the islands, or a group of elite, decided we didn't really, the Moanalos went and the ibis is gone. We really don't want to see the Nene go. Let's put a kapu on that, and then that. And I've looked for evidence that there was a kapu on Nene. Never found it, but maybe that's is what saved them ultimately. Here's an example now from work done by my grad student Taylor Calicrate and other collaborators, Michael Campana. Looking at a genome we sequenced of a Hawaii amakihi a few years ago. And this is just a pattern of variation here. And we also sequenced an Akiapola owl more recently in the last year. And just to give you, this is just two statistics I want you to look at here. Ignore most of this. But the genome length, the genomes of birds are fairly consistent. They're about 1 billion base pairs. GB is gigabases, so the genomes are about 1 billion. A typical mammal is more like 3 to 3.5 billion. So birds have really pared down genomes compared to mammals. And maybe because they fly and they need to be more metabolically efficient, and they don't have as much junk DNA in their genomes as do mammals. The other thing I wanted to point out is the number of what we call SNPs here. These are polymorphisms, single nucleotide polymorphisms, a heterozygous site. So instead of being a CC at a site, it's a CT or some other one. The Hawaii amakihi has almost 4 million of these in its genome. Akiapola is only 1.6. This can be seen here, looking across the whole genome. This is the number of SNPs for every, I think, 10,000 base pairs. And you can see that it's about double in amakihi versus Akiapola owl. So there's a big difference in variation. And if you put this into one of these sophisticated coalescence analyses I mentioned, you actually get an interesting history that can be seen for the two species. So here's the Akiapola owl. It's the red one here, and the blue is the amakihi. The first thing I wanted to point out is, if you go back about 1 million years, you'll see that there's little variation, and then the variation increases more in the amakihi than in Akiapola owl. But it's increasing probably because this is the point roughly the point where the birds colonize from Maui to the Big Island, and their populations are growing. After that point, amakihi stays at a fairly sizable number. These are relative numbers, effective population size. It doesn't mean there are 150 birds. It means that it's a genetically effective number. But you can see it's always about three or four times what the Akiapola owl is. And this makes complete sense because the Akiapola owl is a species that is highly specialized and currently has much lower densities than amakihis, has probably always had lower numbers than amakihis. So the genetics is actually giving us a really good confirmation, or maybe vice versa, of what the ecology and the field is telling us, field work is telling us as well. So that these birds definitely have very different effective population sizes and very different, and this is reflected of their very different ecologies and population histories. That's the second way of figuring out population histories using DNA. I wanted to talk about two examples from our conservation work. The first is some work we've been doing with Dan Gruner and Aaron Rankin to study the diets of rats and birds in the kapukas on the saddle between Mauna Loa and Maunakea. And the question that we came in to ask is, what are the birds and the rats eating? Are the rats eating the birds? And if they are eating the birds, are they as prey, or are they things that they're finding dead, so they're carrion? And so we've been, so Dan and Aaron were getting rat droppings, and also from rats caught in traps up there. And also when they caught birds in mist nets, they would get poop from them as well. And then we would use these DNA methods that we have to try to determine, using what we call DNA metabarcoding, what DNAs were in the poop to tell us what the birds were eating. And we found some interesting results. I mean, first, the introduced birds were having a little bit different diet, Japanese white-eyes and Leothrix, for example, than the native birds. They were eating fewer insects and more plant material than most of the native birds, including the apapani and amakihi and the iwi. The rats, the important thing here is the rats were eating birds, the yellow there, as much as 20% of their diet was bird. And then it led to the second question, are they eating things that have been dead a while, or things that they're finding that they're killing and eating fresh? And so in order to figure that out, I developed an assay to try to see if bacteria in the gut could tell me what they were eating. So we did this chicken McNugget experiment, we called it, where Erin and her students took these, not chicken McNuggets, they just took pieces of chicken that we washed in a light bleach solution and put them out in cages where nothing can get in there but bacteria, or fungus maybe too, but nothing could go in and eat it like an insect. But it was out in the habitat. We replicated that, I think, 10 times around the kapukas. And we let them go, and every certain period, let's see, it was day 1, day 2, day 4, 7, and day 11, we would take a nugget out and freeze it and use it for DNA analysis to figure out the bacteria. So we did what's called a microbiome analysis on this. And we were able to find that there were a lot of bacteria that only showed up early, day 0, day 1, that would indicate that something's fresh. And there were a lot of other bacteria that only showed up late, in days 4, 7, and 9, that would indicate it's something that's been dead for a while. So the bacteria are colonizing and growing, and they're different from the ones that initially colonized. So we had I think we ended up having 9 and 7 biomarkers that looked pretty good for whether something was fresh or something was carrying. Forensic scientists are starting to use this also to determine the age of a body they find somewhere, how long it's been dead, because the bacteria that colonize, like insects, like Lee Goff does with insects, bacteria can indicate the time of death as well. So it worked out really well on that. And then when we did it in some of the actual diets we looked at, where we looked at rat poop that had birds and rat poop that didn't have birds, we were able to find that there were both types in there. So we did find some that looked like they were fresh, maybe fresh killed, and some that were older. So rats are probably eating both carrion, things that are dead for a long time, and also they're probably, we know from observation that rats are taking nestlings and sometimes adult birds as well. So it was a very interesting way to use DNA, though, I thought, to answer these questions. The last thing I want to talk about is the invasive malaria work that we've been doing. And this is a much larger project involving a lot of collaborators here in the islands, including some that are here in Hilo. And like Christina Paxton I'll talk about, and Carter Atkinson, our collaborator up in USGS in Volcano, and also people that are working on the mosquitoes as well. So this system involves native Hawaiian birds, which are mostly susceptible and live at higher elevation, exotic birds that could serve as a source or a reservoir, and then the vector, which is the southern house mosquito, which has been here since 1826, probably introduced from Mexico initially, and then the malaria parasite, Plasmodium relictum, which is present by the 1940s. We found that mortality varies among species. Things like the Avian Laysan finch, they get infected, they die, pretty much. Others, it's more 60-70% die. Low elevation amakihi, if you took and caught an amakihi here that was not exposed to malaria and you exposed it, they would survive. Almost all of them make it. So they appear to have tolerance or resistance to malaria. Same thing with the Hawaiian thrush, the omao. And most of the introduced birds that have been looked at, they will get infected, but they usually won't get very sick and they won't usually die. It's only the native birds, mostly the honey creepers, but some of the other birds also are susceptible, sensitive to malaria. And as I mentioned, the mosquitoes occur at low elevations, and so therefore the malaria and also the pox virus tends to be at low elevations, and that's where you don't find native birds. You'll usually find them above that level, except for amakihis and occasionally papanis at the lower elevations. But it's up here where you find the Achaeopola aus and the eves and the palila. And that's largely because of the mosquitoes and the disease. There's also some habitat issues as well, but it's the disease we think is really limiting the distribution. What kind of malaria got to Hawaii? It's Plasmodium relictum, but it's this particular type called GRW4, which is found mostly in Africa and Asia, and on islands in the Pacific, and also in the Atlantic on Bermuda, and also in the Indian Ocean islands. It's very common on islands. It seems to do very well on islands. And the phylogenetic work we did on malaria parasites shows that the Hawaiian malaria, the GRW4, is most closely related to a set of parasites that come from the old world, mostly from Africa. So we think that the parasite probably came out of Africa and then spread around the world. And we did a lot of ancient DNA work to try to find out when it got to the islands. And the best we can tell is it probably arrived sometime in the 1930s, maybe early 40s. What about the mosquito? The mosquito, I said, was introduced from coastal Mexico by a whaling ship, the Wellington, into Lahaina streams, and then moved rapidly among the islands. But what we found doing genetic work, this is former postdoc Dina Fonseca, and what we found was that the Hawaiian forms now don't really match the Mexican forms genetically, which would be here. Actually more the Australian South Pacific forms of the same mosquito. So the mosquito that was here originally, the Mexican form, essentially got taken over and genetically and swamped out by a new one. And some ancient DNA work on that suggests that that also happened probably in the 30s or 40s. So about the same time that malaria came in. So this we thought might not be a coincidence. So we did some experiments to see if the North American mosquito, Culex ginkgofasciatus, could transmit the malaria as well as the Hawaiian one could. And we found out that even though when we infected them on canaries that were infected with the Plasmodium, when we then let the mosquitoes bite those birds and get infected, the mosquito would get oocysts, oocysts in the abdomen, but those would not develop and pass through the thorax to the salivary glands. So we never got sporozoites in the salivary glands in the American mosquitoes, but we did in the Hawaiian ones. So there was a difference there. The American mosquitoes are not effective at transmitting GRW4. The Hawaiian ones, which are a mix, mostly Australian Pacific mosquitoes, could. So there seems to be a perfect storm here, a perfect match between them. And we've been trying to develop genomic methods to understand this by sequencing genomes and by using those data to look at the populations in the islands and conducting infection experiments. And I have a host of collaborators on this, graduate students like Taylor Calicrate, who worked on the Amakihi genome, and Lauren Sackett, and Christina Paxton, who's here in Hilo, working on challenge experiments and gene expression analyses in response to the infection in Amakihis. And we've also been working a lot on developing Plasmodium genomes and genomic resource mosquitoes, and also on Seca. And we did some looking at the Big Island here, a low elevation Amakihi that survived the malaria, and upper elevation ones that some survive, most don't. And we actually found when we looked at that we across most of the genome, we were actually able to find a number of parts of the genome where we found what we call outliers, where there's evidence of strong selection. And here's 7 of them that are in the circles here. When we looked at those, we found that all but one of those have immune system genes in those regions, whereas you would normally find immune system genes in like 1 out of 50 regions or more. We're finding it in 6 out of 7 of these. So we're now targeting in and trying to figure out more closely what the differences actually are there, and to see if that's what's responsible for why low elevation Amakihi can handle the malaria so well. Christina Paxton and Carter and Lauren and I are also working on doing gene expression analyses to find the same sorts of things. And I just wanted to finish with a take-home message. I have no take-home message. This is sort of a potpourri or a poi talk, Hawaiian bird projects, but I can tell you that we're continuing in the future to do more work on this, more work on honey creepers and their evolutionary relationships, and on other species of birds, and also a lot of work now on the malaria system, including the Amakihis, the parasite, and also on the mosquito. I wanted to just say thank you to all of the postdocs and students that have worked on these various projects with me over the years, because they've really been the drivers of it, and they were great fun to have in my lab, and all the ones I have now are still great fun, and they've really pushed this all forward, and many of them are continuing to work on Hawaiian birds after working in my lab. And I'm trying to encourage as many scientists to work on these problems as possible, because it's really a problem we have here, and I'm not sure we can solve it that easily, but I'm hoping the science will help to solve it, along with better policy, so we can at least have some of these birds still flying around the islands for the next few years. Thank you. And I've gone on a little longer than I think I thought I would, and if there are any questions, by then.
Outliers suggesting there's some resistance among a few individuals.
What it shows is there's genes across the genome that are showing a pattern very different from the background. So those are genes that are probably under strong selection. There could be other genes in there that are also involved, but they're not showing that pattern. So it's what's called a genome-wide association study. GWAS, they call it. It's used for looking at, try to find genes for different diseases, humans.
Does that imply some resistance?
Those are birds at low elevation are resistant. Birds at high are a mix of resistant, and so we're looking for markers. And we're doing it with challenge birds as well. We're looking at the same markers for birds that were challenged with malaria and survived it versus ones that didn't.
So could you breed birds that carry resistance?
That's our hope, is that we'll find the genes that are involved, and then we can find similar genes in the endangered species and try to, in the captive breeding programs, try to select for those. There's also the possibility that we could even do gene transfers. We can actually do synthetic biology and put, if it turns out there's a set of genes, a gene drive, you can use a gene drive. CRISPR method. You don't need a treatment, CRISPR method. You put them in and colleagues I have to do that kind of thing tell me it can actually be pretty easy to do with birds. It's matter of, now you have a bird that's, there's sort of philosophical or ethical arguments about it as well. You have a bird that's, its genome is almost completely still the same thing, but it now has a couple of genes from a related species. So is it the same thing?
Well, it's kind of like what you said earlier, where we collected the samples of birds that are now just gone. And you go, oh, that's terrible, they collected these beautiful rare birds, but now they're gone. And so, it's kind of maybe you have to do that intervention where they're gone.
At this point, I think this is a possible way to help beekeepers down the road. Notifications of mosquitoes would be probably more effective and efficient if you just do it on one mosquito. In this case, you'd have to do it on 10 or 15 species of birds. But I don't want to leave any avenue unexplored. One thing I didn't mention was Kauai is having a crisis. In the last 10 years, I don't know if you've seen that work done by Evan Paxton and other collaborators here at USGS, has found that the birds on Kauai, the Maunakea Swamp, and the state park up there, those birds have been crashing for 10 years because probably evidence really is strong that malaria is not identified. And they've gone down in some species 90%. There were 4,000 birds there 10 years ago, and now there's 400. So, and there's 3 distinct species here that will go extinct in the next 5 or 10 years. And also things like the Avian and Onakiki. Onakiki is a distinct species. Also 4 distinct species.
So many questions. But related to the work that you guys mapped out all the honey creepers and their relation to each other genetically, and you say that they come from an ancestor from Asia. So are there species throughout the Pacific then that are distantly related to honey creepers?
There's nothing. I don't think there's any other cargeline finches across Pacific Islands.
Yeah.
There's nothing that's radiated like that. If you look at a finch, it's pretty much the same thing. A house finch, a rose finch, a canary, they have the conical bill. They vary in color from red to yellow to some green, goldfinches. But they don't show, they still look like a finch. And they have a lot of traits. The taxonomists would say they have a lot of very stereotyped traits that tell them that this is a cargeline. But Hawaii seems to be the only place where cargeline finches have taken off. And I don't think there's any other cargelines in Pacific Islands that have colonized. So, is that answer?
Yeah, thank you. Yeah. I'll raise my hand again for other questions.
Don't talk to me afterwards.
I saw some geese in Sweden, and I wondered if they could be, they look so much like the native, and I was wondering if they could be related.
Which ones?
She saw some geese in Sweden that look very similar to the Nene, if they might be more.
Like barnacle geese? Maybe barnacle geese. And barnacle geese are part of that group. In fact, what's interesting is that, if you look at the tree of Canada geese, you have the large Canada goose, which is now considered a separate species, and the Nene is sister, and the giant goose, they're part of that group. And then the other branch goes to the small Canada goose, and the barnacle goose is sister to that. So they're all part of one group. And barnacle geese do look a lot. They look a little, but I think some people think brant also looks like a Nene, but brant are something that's different. But, yeah.
Rob, there are some pre-act Nene that I've actually seen Nene.
There are lots of native.
Yes, there's
Oh, I'm sorry. Oh, I thought you meant wild geese. Did you mean wild geese or like in a park there?
They're just walking
Oh,
around.
yeah, the barnacle, I used to see barnacle geese in Scandinavia all the time, but I have seen nene in, like Slimbridge has lots of nene. And they are they're the tough geese on the block there. They dominate all the other waterfowl. Slimbridge is a conservation park, a zoo mainly for waterfowl.
I just had a question about the avian malaria. You mentioned people really started noticing it around in the 1940s, but there was a vector right earlier than that, in the late 1800s, early 1900s.
The malaria, a guy named Baldwin, remember his first name? Richard Baldwin, found it he took blood smears of birds from Volcanoes National Park and found malaria parasites in them. So he was the first one to see that. That was I think he found them in the early to mid 40s.
Yeah, but I guess the bigger question I was wondering about in respect to your ancient DNA work is there any indication that there may have been a reservoir of avian malaria already in Hawaii from migratory birds that were going back and forth between different places? Have you guys explored that in the ancient DNA work that you've done?
Yeah, no, that's a really interesting question. We looked for things. We found malaria in a colea, a golden plover, and in a duck.
Huh?
I can't remember which one it was. It's in our paper. But from tissue that we got, the salvaged birds from the Fish Museum, or no, from Fish and Wildlife.
How old were they?
But that was just recent. They were in the last 10, 20 years. We also looked at some old museum specimens of introduced birds and didn't find any malaria parasites in them before any at all. And we also, but we haven't looked extensively. I never thought to look at plovers, museum specimens of plovers, go back 100 years or so. But there probably were malarias that were coming in with plovers or other shorebirds or with ducks, but there wasn't the right vector here,
Right.
the right mosquito to take them and move them into the native birds. That's pretty common in a lot of cases. That's why we don't think the GRW4 is very common in the Americas, is because it doesn't have the right vectors.
So you said that the mosquitoes came from Mexico and then Australia, but the malaria, the plasmodium itself, came from Africa.
Right.
Was that brought over from Africa through a mosquito or through an African bird? Or
Yeah, we don't know what it came from. Whether malaria actually, we found the same lineage of malaria in French Polynesia. It's been found in New Zealand, found in India. It's spread around the world. The only reason we think it came out of Africa is because of the tree showing that its closest relatives are in Africa. But it could have come from India or Australia or somewhere. But in terms of evolutionarily, it's out of Africa. But in terms of where it came to Hawaii from, it's hard to say. And I've looked at probably 150 specimens of introduced birds to Hawaii to screen them for malaria, and I've never found any. But they were all from the early 1900s, and I never found any malaria. We did have one case, we've never been able to replicate it, of another lineage of malaria showing up in a Hawaiian bird, an early Hawaiian bird, but it's not the GRW4, it's something very different from that. And so we think it might have been one of those rare contaminants that we get sometimes in labs. But we tried replicating it several times. I even sent a sample to here for Sue to work on. I even sent a sample of it to San Francisco for a former postdoc to work on, and they couldn't get it to come up either. So I think that's pretty good. But I've always wondered if that's the case. If we look at these other things, we'll find something else for us here.
mitigation is an issue in terms of protecting the birds, because mosquitoes can reproduce at the higher elevation and exist at higher elevations, then what are the implications of global warming, and are we in danger of losing these birds because of global warming?
Probably should say global warming does not actually occur and is not man-made. It's a big implication. We think that the mosquitoes are moving upslope because of global warming already. They were much lower in the time when Richard Warner found malaria, studying malaria in the 60s here, and Charlie Van Riper. And then Carter Atkinson and his colleagues at USGS have documented the rise, increase in the Big Island here.
And do you see that being related to the incidents on Hawaii?
We think that's what happened on Hawaii, that probably just the climate was shifted. Also, there could be adaptation by the mosquitoes to get, and then one other thing is that the Culex fasciatus from Mexico, from North America, tends to be a mosquito that's warm weather adapted. It lives in Mexico, it lives in southern Florida. They get up to Washington and they hybridize with Culex pipiens there. And so they don't do well in the cold. Whereas the ones that come from the South Pacific, the Australopacific kind of Culex, they're in New Zealand at higher elevations. So this switch over from the Mexican type to the Australian Pacific type might also have led to an increase in upward slope. But we don't know if it's both of those factors or one, probably both of them. But definitely, as the planet warms, as climate shift, as things are, this zone of habitat is going to be diminishing. Vegetation can go up, and to move the zone upwards even more, but that, it doesn't happen as quickly as mosquitoes coming in. 10 years, what they've done.
So I got a little clarification. You had one slide with 2 species of birds, and with that genetic analysis of DNA, you could estimate populations.
Right, that's the amazing thing about these. So you look at the pattern. If you look at the patterns of variability in the DNA, because a population size that's larger will allow more genetic variability to accumulate than a smaller population. And the pattern is one more of selection in the larger populations and one more of what we call drift or random processes in the smaller ones. It's like throwing dice. If you throw it 5 times, you might, or flipping coins. If you flip it 2 times, you might get 2 heads or 2 tails, or you might get 1 of each. If you flip it 10 times, it's more likely to come closer to the 50-50. So the random processes of chance processes operate more in the smaller populations, and this determines what the pattern of DNA variation. You tend to get more mutations, what we call fixing, in the larger populations. I'm sorry, more fixing in the smaller populations than the larger ones. So mutations can go that. And so the pattern you see in the variability, you can actually model what the population would be like. Now there's standard errors on that. You saw that in there, but if you have enough data, the standard errors get really, really small. And at least and you might have noticed that the standard errors were very small. That sort of cloud of points on that figure were very small further back than they were closer up, because you have a lot more data to tell you what was going on before than you do what was happening recently. So we really don't trust what was happening in the last 10,000 years from those graphs. We trust more what was happening, to a million years back. But it's, I can, give you some references that might explain it. But coalescence modeling has been used a lot in the last 20 years or so in genetics, and they've reached a really incredible level of sophistication. It's better done with more samples. And, if you had 10 genomes from Akis and 10 genomes from Amakis, you'd get even better. But even from 1 genome of each, you can get 3% results. We'll see how they match when we get